Hi, my name is Beth Corelli, and today I will be presenting microbiome individuality and IBD, how personalized guts shape disease. Inflammatory bowel disease, which includes Crohn's disease and ulcerative colitis, is a chronic inflammatory condition of the gut. Unlike infections caused by a specific microbe, IBD is multifactorial, driven by genetic susceptibility, environmental exposures, immune dysregulation, and a disrupted gut microbiome. Here's a visual of the parts of the large intestine that Crohn's disease and ulcerative colitis respectively affect. In a healthy gut, trillions of microbes live in a balanced, mutualistic relationship with the host. They support digestion, train the immune system, and produce metabolites such as short-chain fatty acids that strengthen the epithelial barrier. Every person's microbiome is unique. Only about 1/3 of gut microbial species are shared between individuals. Even identical twins, who share nearly all of their DNA, have very distinct gut microbiomes. That individuality plays a major role in how people respond to disease, diet, and drugs. The host shapes the microbiome through factors like immune signaling, mucus composition, and antimicrobial peptides. For example, variants in genes such as NOD2, one of the strongest genetic risk factors for Crohn's disease, alter microbial recognition and disrupt microbial balance. The environment also continuously reshapes the gut microbiome. Diet is one of the strongest forces. High fiber diets promote beneficial short chain fatty acid producers, while Western diets reduce microbial diversity. Antibiotics, early life exposures, and stress also play major roles. Microbes also interact with each other. Beneficial bacteria produce butyrate, a molecule that fuels colon cells and suppresses inflammation. When diet or immunity changes, pathobionts, normally harmless residents, can expand and contribute to disease. In IBD, the relationship between the host and microbiome collapses. Beneficial bacteria decline while inflammatory bacteria increase. A weakened epithelial barrier allows microbial products to enter the tissue and activate immune responses that fail to shut off. The immune system constantly samples gut microbes through receptors like TLRs and NOD-like receptors. In IBD, this process becomes dysregulated, leading to chronic activation of the Th17 pathways and pro-inflammatory cytokines such as TNF alpha. Evidence shows that microbiome disruptions can precede disease onset. In mouse models, transferring the microbiome from an IBD patient into germ-free mice can trigger intestinal inflammation. Even in humans, the success of fecal microbiota transplantation depends on whether donors can carry key beneficial microbes. Because every microbiome is different, patients vary in their response to the same treatment. This is driving interest in microbiome-based precision medicine, where therapies are tailored to the biology of the patient's gut ecosystem. Instead of trying to eliminate bacteria, future therapies aim to restore microbial function. That includes postbiotics, delivering microbial metabolites directly, and defined microbial consortia as a more controlled alternative to fecal transplants. In summary, the gut microbiome is highly individualized, and IBD often develops when communication between those host and microbes breaks down. Recognizing this individuality is key to understanding why IBD is so variable and to designing therapies built around the biology of each patient. Here are my references, and thank you for listening.